Good evening to you all. I'm Michael Adubuache. So if you're meeting for the very first time, that's my name, and we'll be going through continue with our lesson on the classification. Since last week we started classification, we talk about the forms of classification, the cladistic, the hierarchical, the phylogenetic, and also the numerical. Then we look at Aristotle's mode of classification. Then from there we talk about the hierarchical classification by Carolus Linnaeus. Then just last Sunday, we look into the types of classification, particularly the natural and artificial classification. Then we are to continue on from how the binomial nomenclature is used and the rules. So that was where we ended. So since we are looking at classification of lower organism, we will now just dive into how to classify them. So we use amoeba as an example or a case study for our classification along the taxa that Carolus Linnaeus brought in a system of classification. So this evening, okay, my voice is loud. Try and project a little bit higher. So today we'll be looking at the life processes, the structure and economic importance of amoeba proteus. So that is what we are going to look at this evening. So is there any question from classification up till now you can ask? If there's a peculiar question, something you want us to deliberate on, or you want to draw attention to from last week's lesson, let us know so we can start. I'm waiting for just 30 seconds. If there are no questions or concerns, we will be good to start today's lesson. Alright, no hands up yet, so let's start today's lesson. So as we can see from my screen here, we are to talk about the life processes and the economic importance of amoeba proteus. Zedagwe Bright Sarah says she can't hear. Is there anyone else who can hear my voice? You can give me a thumbs up. If you can't hear my voice, give me a thumbs down. If you can hear me, give me a thumbs up so that we will know that Zadabui, Mabel can hear me. Lenient, okay, Janet Boache, Stephen Kwate, I think the regular and the punctual members can hear me. So Zadabui, if you can't hear me, check your network. I think today is the first time I'm seeing that name from the list. Natalia too, you've been very, very punctual and you always come on time. So that way, kindly check your network. You can decide to leave and join the meeting later on. Okay, so pardon me if I can't raise my voice that loud, so at least I'm sure if you can increase the volume on your device, you can hear me very, very well. Alright, so let's continue our lesson. So as I earlier said, today we are looking at the life processes, the structure and the economic importance of amoeba proteus. So we remember in the level of organization, we have cell, then from cell we move to tissues, from tissues to organs, organs to system, then system to the organism. But some cell can jump the tissue organization, the organ, the system before organism, and that single cell can serve as an organism. The reason is only that cell will be capable of performing all the 7 life processes. And if that single cell can perform all of these processes, then it qualifies to be an organism. And such organisms are called single-celled organisms. So these are the organisms we are coming to talk about. We will talk about 3 of them in the first year, and in your second year we will talk about some others or the remaining in our syllables. So we are looking at amoeba proteus, and mostly that's the first unicellular organism you will learn, and it's one of the simplest life forms that we can have now. So that is amoeba proteus. So the scientific name, as from the Carolus Linnaeus binomial system or nomenclature, is Amoeba proteus. So yesterday we look at from the kingdom up to the species of Amoeba proteus. You can refer to the video that I posted today, that is classification lesson 3. You're able to see all those things that we learned last Sunday. So Amoeba is the genus name and the proteus is the species name. So by conversion, if this is to be the scientific name as we are written, this amoeba, this is not the actual conversion of the scientific name, rather this, but this should be underlined. So this should have been underlined and the proteus to underlined separately. So if you are writing a scientific name, this should be underlined or rather be italicized. So it should be underlined separately. So let's keep that in mind. Now, as I explained earlier on, amoeba is a unicellular organism. Uni means one, cellular means a cell already. So it's a single cell organism, just one cell capable of performing all the seven life processes. But humans, we are multicellular. We have millions and billions of cells in our body. They work together at some level to form tissues. Some come together to form organs. Then we have a system. Then putting all these systems together, we have an organism that is capable of performing life processes. So amoeba is a simple and one of the simplest unicellular organism, and it's in the kingdom protoctista, as we know already, or we can say kingdom protista. Instead of protoctista, you can simply say kingdom protista. And the phylum of amoeba is rhizopoda, and we can say Sarcodina, but Sarcodina is a primitive, or let me say an old name. Some will say an archaic. So it's called Rhizopoda, that is a phylum, or we can call Sarcodina. These can work, but this is now the contemporary phylum we give it to it. So let's look at the size of amoeba so that we can appreciate how small it is. Now look at this measurement, millimeter. It's not a centimeter. We know a centimeter is something small. So this one is a tenth of a centimeter divided into a quarter. So meaning 1 centimeter divided by 40. So look at your rule, just check for 1 centimeter divided by 40 times. That is the size of amoeba. So it's like just a pinhead or something is smaller than that, a speck of a powder or from a chalk grounded into powdery form. Very, very, very small. So it is about 0.25 millimeter. And we know a tenth of a centimeter is a millimeter. And we have divided it by 4. So 4 times 10, 40. So just 1 centimeter divided by 40. And that will be the size of amoeba. So from 40 to 20 divisions of a centimeter, that is the size of amoeba protein. Very, very small to be noticed. And even if it was to be colored, you will be able to see it. But since it's very, very transparent, it's very, very difficult to see amoeba with the naked eye unless you have some stents or you check from the microscope. So the length is 0.25 millimeter to 0.5 millimeter, and it can only be seen with a lens or a microscope. So hardly can you see it with your naked eye. And the shape of amoeba is irregular, so the size is very minute, 0.25 millimeter to 0.5 millimeter, and the shape is irregular. It doesn't have any definition, so amoeba can assume any shape. So I used to tell my student that when we are drawing amoeba, there are no mistakes about the shape or the how it looks like. Just that the line should be faint and sharp and not clear and not blurry. So once your outline are clear, you are good to go. That is an amoeba. So it is irregularly shaped, and we will explain why it is irregularly shaped, because it has only a cell membrane and it moves. The way it moves, the cytoplasmic content can move anyway, anyhow. So based on the direction of the movement, it can assume any shape. So amoeba is irregularly shaped because the cell membrane is very, very thin, flexible, and is very, very fluid. So it can move and assume any shape at any point in time. So that is the structure, the shape and length of amoeba proteus. Now let's look at the body. The body consists of a cell membrane, I've talked about that already, and the protoplasm. When we say protoplasm, there is a word you can see from it, which is cytoplasm. And so cytoplasm plus the nucleus form the protoplasm. So protoplasm is like the nucleus and the cytoplasm and everything. So when you take the cell membrane away, what do we have rest is the cytoplasm, that is, let me say, the fluid or the solvent in which the nucleus and all the other organelles are dissolved into. That becomes a protoplasm. So the nucleus plus cytoplasm form the protoplasm, or someone can say the cytoplasmic content and everything over there becomes the protoplasm. So the protoplasm is surrounded by a cell membrane, that is the everything inside within is the protoplasm, and it is being surrounded by a cell membrane. Now the cytoplasm that we know is like the fluid or the ground substance of the cell is made up of two distinct zones. So we have an inner layer and an outer layer. So the outer zone is called ectoplasm. So ecto is a prefix that means outside. So ectodem, that is a part of the skin that is outside. So ectoplasm or plasma gel. Then we have the inner part, and the outer zone is thin, clear, and jelly-like because it forms part of closer to the cell membrane. So it's a thin, clear, and jelly-like fluid that is able to move, that gives its fluidity. Then we talk about cyclosis very soon. Now the inner zone is also called endoplasm. Endo means inside, so endo, like endocap, that is a part of the seed inside, the inner part of the seed. And the endoplasm is also called plasma cell. So plasma gel is the outside, jelly like. So plasma gel, jelly like. Use that mnemonics or help you able to remember. So plasma gel is the outer part, is jelly like because it's a plasma gel, jelly like. Then we have the inside plasma cell. So cytosol, or that's the inner part where it's able to move. So the inner zone is called endoplasm and also called plasma cell. It is dark and more fluid. So the inner part is very dark and more fluid. So these are the distinct zones of the cytoplasm of an amoeba. Now, amoeba proteus. So there's a better way to write the scientific name. Here it is being italicized since it is a print. We could have underlined it if we are writing it in ink. So amoeba proteus is a protozoan. So it's a protozoan and it's a protozoa. These are types of free living microscopic organisms. So they are protozoa. Some are also bacteria are there, they are on their own. So this one is protozoa, and some are other parasitic and could be very, very harmful. So protozoa are generally free living unicellular organisms, and amoeba is a protozoan commonly found on the bottom mat. So let's look at its habitat. It's usually found under the bottom of a mud or a pond, or the underside vegetation in fresh water. It could be found in ponds, ditches, lakes, springs, and slow running streams. So normally, since you look at a structure, it's like putting a cotton wool inside water, like a cotton wool that has been opened up, that it looks very, very soft. So looking at that structure, it can't stay in a fast moving water like the river, a fast flowing water. It can't live over there because that can distract or kill even the cell because it's very, very soft and very, very fluid. So it lives in either stagnant or slow moving water body. That is why amoeba can stay in such a environment. So it is found mostly in comparatively clean ponds. So when the pound is very, very clean, amoeba could be there. So it is found in most in comparatively clean ponds with extremely oxygenated fresh water. It can also be found in a large food web ecosystem with other organisms. So it can survive whether in a clean environment where there are no competitors or other organisms in the ecosystem, or even in a very crowded ecosystem. Amoeba can also have a place in such an environment. So it's also found in large food ecosystem that incorporates many algae and plants. So algae too can serve as the producers in the ecosystem, as amoeba can also start as the as part of the system, but they are not largely not producers. They are rather consumers because they rely on food and other bacterial organisms in the water. So it is also sheltered under the size of lotus pond. So these are where it could be found. Maybe lotus pond, it could also be found there, and it moves and feeds with pseudopodia. So I could recount one of our meetings, I think 2 weeks ago, someone asked a question about pseudopodia, and I told the person that there will be a day we will talk about pseudopodia, but I'm sure she got a very good explanation. So pseudo, as I explained the other time, means false. So pseudonym means a false name. So if maybe I'm writing a story and the person is maybe Mabel, but I want to make it Akushika just to cover up the name, then the Akushika I've given to Mabel is her pseudonym. Then we also have something we call a pseudocode. The IT people will know much about that. That is a false code, or maybe something that's an actual coding for the program or instruction they are doing. So pseudo means false, and podia from the word pod, which means feet. So pseudopodio means false leg or false feet. So we have it here. Pseudos means false, and the podus, that's the actual Latin word, which means leg or feet. So amoeba doesn't have a locomotory structure. Mind you, animals like we humans, we have our legs, some will say limbs in other mammals as our locomotory structure. The birds and fowls have the wings for locomotion. Other organisms have flagella like the bacteria, even the euglena has a flagella, but amoeba doesn't have any locomotory structure. It doesn't have any structure that allow it to move from one point to another point. When we say locomotion, it means moving the entire body on your own from one point to another point. So right now I am not locomoting, I'm just moving my hand. But if I move from this point and I go to the other side, whereby I will not be seen in the camera, I have locomoted because my entire body has moved from one point to another point. Amoeba doesn't have such a structure, but what they are able to do is by moving their cell membrane and their cytoplasmic content in a specific direction which allow it to move. That is how amoeba moves. So it has pseudopodia as its false locomotive structure, but in the actual sense, they have no structure or external structure or maybe let me say peripheral structure attached to their body that allow them to move from one point to another. I will explain that very shortly for you to understand when we reach the movement of amoeba. So this is the structure and the drawing of amoeba. So all that I was talking about, so the entire structure that we see here, we have zoomed it under the compound light microscope. So this is how amoeba could be seen under the 40 times microscope power lens. So all of these will be around lesser than 0.5 millimeter. So we have the pseudopod. One is called pseudopod. So one is called pseudopodium. That is the actual name. So here it's written pseudopod, but one is called pseudopodium. Then when this we have many, we call it pseudopodia. So this is a pseudopodium. Then another one is here. This will be one. This was some that was formed some time ago. So we can have 1, 2, 3, 4, 5, 6. So meaning the amoeba would have moved in all of these directions. But currently, how will you know the direction is moving in? The longest or the lengthiest one. So it could be either this point or that point. The reason is, as you see each of the projection, there's a food vacuum there. So this suppose that the amoeba moved or extended the cytoplasm here to feed on this food. It also did one here to feed on this. Then here it also fed on the food. Here too it fed on the food, and another one here. So looking at how close it is, that is why they have named it the pseudopod. This would have been the last movement of the amoeba. The reason being that this vacuole is closer to the cell membrane than any of them, and even the drawing named here as the pseudopod instead of the rest, although they all used to be a pseudopod that amoeba used to move. Later on it will try and retract or withdraw it back. So you see this one supposedly was from here, but now you see that this one is very very short. It is now trying to, it will withdraw this one until it becomes flattened. Then that will be the end of that pseudopodium. Then it assumes its normal cell membrane structure. So that's the pseudopod. Now we have the nucleus that is always normally in the center of the cell. Then this one is a contractile vacuole. So contractile vacuole is a water vacuole. So when we say contractile vacuole, is a structure in unicellular organisms, mostly the proteus that allow them to balance water. So it is for osmoregulation. Remember, from osmosis, osmo refers to water. So when they are trying to balance water in their body compared to the external, we call that one water balance or osmoregulation. So contractile vacuum is a structure that is in this organism for water balance, and that is why it is here. Then you can have a lot of the food vacuums based on the food that it will phagocytosize. So a food that it will engulf will be here as the food vacuum. That's the contractile vacuum here. Now we have the ectoplasm, as we explained, is thin clear jelly-like, and the inner part is dark fluid filled, and that is called the endoplasm. Now all these are food vacuums, as you see here, the orange with black structures inside. All these are food vacuums, and these are crystals that is found in the body of the amoeba. So that's the plasma membrane, or the cell membrane. It's very fluid, and the reason it can extend so that these structures in the amoeba or the fluid in it can move. As they move towards the end of the cell membrane, it causes it to extend, and it causes what we call a pseudopodium or an extension. I will pause at this point. If there's a question, you can ask. If you have a, if you have a question at this point, you can raise up your hand, and I will invite you to talk. If there any question, you can raise up your hand, then you ask. If you understand too, you can give me a thumbs up, then we can move on. If you have a question, you can raise up your hand. Okay, Divine Chira, I can hear, I can see Janet Boache, Nanama. I think a hand just came up. Yes, Stephen Quatin, you can ask your question, Stephen.
Okay, sir, thank you.
You're welcome.
You said amoeba is a unicellular organism,
Yes, please.
but in my book, it says lower organisms such as amoeba, iguana, spirogyra are single-celled organisms, multicellular or multicellular.
Mention all the organism that you mention the organism again, and I'll show you something there.
Amoeba,
Mm-hmm.
iguana,
Okay.
and spirogyra.
Yeah. Very good. All these organisms are even unicellular organisms. But one thing about Spirogyra is unicellular organism, but they live in filamentous form. Oh, don't worry. How they move, we are about to start it, Natalia, so don't worry. I want you to understand the structure before we talk about life. So very soon we will talk about how it moves. It's very simple. So you get to understand it. So let me come back to Steven's question. So all of them, we say lower organism. Why are they lower? Because they are smaller. Like they are way below the multicellular. So low organism there also means unicellular organism. Remember the book also said single cell. Single means uni. That is one. So it's the same thing, just that we are using different diction. And the reason why I'm pause here is that Spirogyra is unicellular organism, but Spirogyra exists as filamentous form. So it's like taking what do you call this one? Sugar cane. You see, sugar cane, we have different points that we call the node and the internode. When we take one part of the sugar cane, the internode, that point is like one Spirogyra. It's a single cell. When you take another internode, it's another a single cell. You see that when you take one sugar cane, you can have about 10 or 15 of those internodes. That is how Spirogyra looks like. We are not looking at Spirogyra today, so I will not dive in much. So let me use that to explain to you. So each of the node in the sugar cane is also synonymous to a Spirogyra cell in their filament. So they exist in a filamentous form. So they are attached together and they live together. So when you take one pack of Spirogyra from the ground, or maybe from the wall, though they are single cell organisms, but we have them in several compartments. So, but we don't call it a multicellular organism. The reason is each of them live on its own. They are independent. It's like living in a rented apartment. That I'm in my apartment. I do everything. That I don't share anything with the other person. So if it's a compound house, we all use together. So that would be like a multicellular organism. But if I have my own apartment, I do everything within my place, I might not even need to come to your place, then that becomes unicellular. I'm just solitary person. So Spirogyra is also a cellular organism, but a compartment or a filamentous form put them together, but each of them is capable of doing its own life processes. So all of them are single cell organisms. Steven, I hope this should make you okay. I've been gone a little bit to explain further for you. Yeah, so the unicellular organism. Later we talk about multicellular organism. Okay, I think a hand came up. Whether it was Patricia, Jesse, a hand came up. Can that person ask the question? Yeah, Patricia, your hand is still up. If you can hear me, Patricia, ask your question.
Okay, sir. Yes, the other day you were talking about the phylum that it's concerned only plants. So that means amoeba falls under phylum.
I know, ask the question again.
The other day you were talking about phylum as it's also called division under plants.
Yeah.
So that means amoeba falls under phylum.
Yeah, since amoeba is a prototype, its phylum or its second taxa is phylum. You will not call it division. Are we getting it? So there are seven taxa. First one is kingdom, second is phylum, but the phylum is well said to be division for only kingdom plantae. So since amoeba is a prototype, is not under kingdom plantae, then we will say the second taxa under kingdom protoctista is phylum. So we will say there's the phylum. The second taxa of amoeba is phylum, not division. That is just as you said. Now, Stephen, as I was explaining to you, this is what I was talking about. You see, this is like the node, another node. This one is a single Spirogyra cell. This one's another single Spirogyra cell, another one. So it's like living in an apartment. This one doesn't share anything with the other one. So here they are undergoing conjugation. So this one is like an individual on its own, another individual. So nothing here moves over there. So that is why we say that they are unicellular organisms, but they exist in filamentous form. Now let me chip in this thing. When I was talking about the life organization of life, I talk about cell, tissue, organ, system, and organism. Now there are forms in which cells can exist, and I think there are four forms. One, a cell can exist on its own, and we will call it a unicellular organism or a single cell organism. That is the first form. Or the cell will exist with other cells and it will become a multicellular organism. So either the cell will live on its own solitary, then it's a cell organism, or it will form part of an organism. That is the first two. Some two, they are single cell, but they will live as colony. They will live together. Example is volvos. Though they are single cell, but they don't live alone. Someone will say they are like Liverpool. They will never work alone. So they come together and they live in community. We call them, they live in colony. Example is the volvos. We have others. They are single cell, but they will live together as filament. Example is what you are seeing right now. That's the Spirogyra. So either you are single cell, living a solitary life, so single and free living, single but living in a colony, single but living in a filamentous form as Spirogyra, or you are part of a living organism. So the palisade cell is a part of the plant cell, or sorry, is a part of the plant, and the red blood cell is a part of the human body, particularly the blood tissue, the blood. Then the Chlamydomonas is a unicellular organism, but it lives together with other Chlamydomonas. Volvos to live with other volvos in a community. Then Spirogyra cell will live in a filament or in attachment alongside other Spirogyra strand. So that is the answer I want to give. Any other question before we can move on? Someone said she wants to understand the movement. So if there are no questions, I will just move straight to the movement of amoeba, that is its life processes. I'm waiting for just 20 seconds. If there are no questions or reactions, I'll continue. Okay, so Natalia says, say about how they move, I do not understand. Don't worry, you understand. I don't know whether you read and you still couldn't comprehend. We are here to explain to everyone's understanding. Alright, so let me continue. If there are some questions later on, you can raise up your hand for me to call you, or you can type it in the message section. Alright, so that is the structure of amoeba proteus. Let's look at the life processes. So the first one we will talk about is the movement. How does amoeba proteus move? So as I said earlier on, amoeba doesn't have a locomotory structure, but what it do have is the cell membrane that is able to extend and allow it to move. So amoeba move by forming what we call a pseudopodia, singular pseudopodium, that is called a force feet. So, and the movement is called cyclosis. That is a slow streaming movement. We call it cyclosis. Now, let me use this as an example. I was looking for something around my vicinity to explain to you. If you have a bag, maybe a jute bag or a sack, and you put something into it, and that thing tilts to the left side, what happens to the bag? Someone should help me to explain. You have a jute bag or a sack, and you have packed maybe cassava or some good into it. If what you have packed into the bag or sack tilt to the left side, what will happen to the bag, or what will... Okay, this one I want someone to answer me. So someone unmute your mic and talk to me. Oh, let's hurry up. Normally, if I keep on talking, it doesn't become interactive. But if I want you to talk to you, you don't come up very fast. This is a simple question. Anyone can answer. Please, one person, open up your mic and talk to me. What will happen? Just a real life application. Okay, someone said the question again. So please be listening to me. Very good. Someone said the bag will shift to that side. So the question was, you have a jute bag or a sack. You have packed things inside, maybe your shoes, anything that you have, cassava or yam. You went to the farm or the market, you have packed things in it. So if the cassava shifts to the left side, Natalia answered that yes, the bag or the sack will shift to that side. So that is what happens to amoeba. So amoeba is like maybe very good. When we were kids, we used to do this. I wish I would have done that. I want to get more idea about the contractor vacuum. Okay, I'll explain later. Contractor vacuum. Alright, so when we were kids, when we buy this, what do you call it? We call it, is it chichiri water? That's why we're saying it. But now it's just the sachet water. In our time, you go and buy, we call it pagini. They'll put it in that white rubber, they will tie it. Then we can pull one side at the top. You pull the other side. Then we say it become like a, how do you call it? Like a toy. Then sometimes you can even squeeze it, you can give it a belly, then we can do a lot of things. Now what happens, because the rubber is a little bit flexible, when you remove one side it can form a tail, it can even form a horn, it can form an eye, a mouth, you can even get a belly out of it. Then you see that, hey, my water is pregnant, my toy is pregnant. Now why were we able to do that? Because it was very flexible. When you extend it, what happened, the water will move to that side, then it will form whatever thing you want to form. That is how the pseudopodia is formed in amoeba. So amoeba is having a flexible cell membrane, that is the plasma membrane. Now it has what we call cytoplasm or cytoplasmic content, the nucleus and any other thing in this body. Now if you extend it or you stretch it, the water in this, or not water, sorry, the cytoplasm, let me say protoplasm, so everything together in the cell is protoplasm, in the cell membrane is a protoplasm. So this cytoplasmic content or the protoplasm will move to fill the vacuum that you have created or the space you have created, and that will cause the amoeba to move to that side. I think this one now will not be a good thing for me. So as I'm talking about the sack issue, if anything that you put into the sack, then it will shift to that side. As it shifts to that side, what happens is it propels or compel the amoeba to move towards that direction. This, let me use this tissue paper as an example. It is torn, so I couldn't use it very well. So assuming this is our container or our sack. So this one is well packed, and this one is the cytoplasmic content. Together it's called protoplasm, and this is the cell membrane. Now what happens if I move the tissue to this side? What happens that the cell will begin to move to that side. Here becomes protruded and extended. Here become what constricted. Then the amoeba will move towards that side. So that's why I told you that when you put your items into your bag, sometimes you see that the bag will tell that way because you place your lunchbox at that side. Maybe you put your dictionary, voluminous book, so you see that that part of the bag will be extended. So that will be like a pseudopodia formed by the amoeba. So what the amoeba does, if amoeba want to move, it will push cytoplasm. I know you know. So cytoplasm is like the ground, let me say the solvent of the cell. When you enter the cell, let me use as example. Maybe you have prepared a soup. The pot is the cell wall or the cell membrane. Or let me use as a cell membrane. So the pot is the cell membrane. Simple as that. Because every cell has a cell membrane. Now, the soup itself becomes the cytoplasm. Now, if there is a meat over there, maybe the bigger, let me like a bowl of fufu with let me say soup on it. Let me do that one. So your bowl will be the cell membrane. Now the fufu inside will be the nucleus. In plant, the larger one will be the vacuole, but now let me use as animal, so it will be the nucleus. Now we see that there's a soup around, so the soup itself becomes the cytoplasm. We have meat inside, so depend and okra. This will be the organelles inside your cell. Now the cytoplasm is like the ground substance, like the solvent or the fluid in which all the other cell compartment or organelles are suspended inside. So if you take the sea, the water becomes the cytoplasm. We have shark inside. Let me use the shark as the nucleus. We have maybe dolphins. Dolphins are the vacuoles. We have small fish over there. They are the mitochondria, cytoplasm, the rest. So the cytoplasm is like the ground substance or the fluid or solvent in which all the other cellular components are suspended in. I'm sure you understand the cytoplasm. Now, protoplasm is the nucleus plus cytoplasm. So it's like if you take a cell and you take off the cell membrane, so like if I take the bone, whatever remains become my protoplasm. So it's the cytoplasm and everything dissolved in it plus the nucleus. So nucleus plus cytoplasm is equal to protoplasm. So I'm sure this one should help you understand. So someone has said something here. Very good. So I think that's very, very important. Let us see what our friend shares. So go to the message section. Someone just showed something there. So protoplasm is the entire living substance inside the cell. That is everything. And cytoplasm is a daily-like substance. That is it. So that's what I just explained. So Lina Fuye, thank you for sharing this information with your colleagues. So you can check the message section. Our sister Lillian has provided it over there. So some of these things I'm sure, but at least it's good they do ask. So it's good sometimes you ask some of these questions that to clear your doubt. So now let me go back to what I was trying to explain to you all. So in the movement, the amoeba will now, so now I can freely say cytoplasm and protoplasm because we all understand. So amoeba will now push its cytoplasmic content. If I say cytoplasmic content, I'm talking about the protoplasm. Everything there moves, so it will move them towards that direction. Amoeba has senses because it's an organism. It senses irritability. So when the amoeba senses a food, maybe this is the food that amoeba has sensed. Now what you do is that the cell membrane will now move. And how can the cell membrane move? That is the cytoplasmic content will now push towards the side. And it's very slow thing. It's not a fast thing. we call it cyclosis, a slow streaming movement. So it will move slowly towards the side. It's like pouring water on a table that looks a little bit slopey. So the water moves slowly, it doesn't go very fast. So the protoplasmic content will move towards, and as it moves towards that side of the cell membrane, you see that that place become extended, and the cytoplasm will move towards that side. So it will call the extension of the cell membrane, and amoeba will move towards that side. Now it will open up, and we call that part pseudopodia. So let me show you something from the drawing here. So this is the amoeba. So the amoeba, let me draw something for you. The amoeba would have been something like this. So please kindly watch. Amoeba can be any shape. And there's a food here. Let me change the color of the food. So this is the color of the food. So amoeba has sensed a food there. Now what will the amoeba do? And there is the nucleus, and it has the contractile vacuole. Someone asked about contractile vacuole. Let me use this point to explain. Contractile vacuole is like a water storage system. What it does that it store water and it maintain water balance. We know that humans, we drink water, our kidney will do that for us to maintain a water balance, and over there, them they don't have kidney. What they have is a contractile vacuole. So they have food vacuole. So the food vacuole is like their alimentary canal, and the contractile vacuole is like their kidney. So what they do is that their contractile vacuole takes in water into the amoeba. If it loses water, if there's enough water in the amoeba, it will also leave. I'll explain that shortly when we reach excretion, so I'll not go there. So contractile vacuole is a structure, very extensible, and that stores water, and it controls osmoregulation, so that the amoeba will not lose too much water, and the amoeba will also not gain too much water. I will explain that very shortly. So this is the contractile. Now amoeba has seen the food here. What it will do is the amoeba will now start to move its cytoplasmic content. So everything within the cell, so all the structure here will now begin to move. So they will move, the structures will now move towards the side. So that will be how the cell. So if all the structures in the cell or the protoplasm begin to move, now something will happen. It will cause the cell membrane to extend. So now the cell membrane will now begin to extend. It will become something like this now, and still it will still be pushing. So let me change the colors as I talk about. So and now this our cell nucleus here will now be somewhere here. It still hasn't reached that side. It will continue pushing it. Everything continue be pushing. Then it will form a structure like this, which is now about to engulf the nucleus. It will continue moving till it completely engulfs it. Now this will become our new amoeba. So I want to erase it. Our new amoeba will now be like this. Since it has eaten everything now, our new amoeba will now become something like this. And now it has taken the food. So let me redraw it again because I drew that one continually. So this will become our new amoeba. It has taken up this food. If you see another food here, it will do a similar thing. Then it will also engulf it, then to become part of it. So that is how the amoeba moves. So it moves by pushing its protoplasm or cytoplasmic content. As it pushes that side, the cell membrane will extend. So if you are in the house, just get some polybag and tie it. Now just tear it. If you have that rubber, do what I'm talking about. If you do that thing, if you extend the rubber, you see that the water will move towards that side, and that's how amoeba moves, by moving its cytoplasmic content. How did the sachet or the rubber get a life? How did they get a feed? We were kids, we were doing all these things. So you just tear one side of the rubber, it's hey, have a head, and sometimes we even do phallus and the rest. And see that, hey, my toy is urinating. All these things we were doing it as kids. So at least that was also a way of depicting how amoeba moves and does a lot of things. If you have a question still on this, let me know. I'm sure this should help you understand, but maybe someone who just joined will be complaining she doesn't understand. If you have a question only on movement, The other questions I will later go there because that will be confusing us. So let me finish. When we are done, I'll go to the comment section or for questioning time. So only type your questions when I open for questions. So you can type it, copy it, then you post it only when we are ready so that it doesn't drag us back. Okay. You can raise up your hand if you have a question. I'll just look on and answer you. A hand just came up. Yeah, Patricia, just say, ask your question. Patricia, you are first, so ask your question first.
Okay, say, does the amoeba move to create a duplicate?
As in moving, then another one as well will be formed.
Yes.
No, it doesn't move to form a duplicate, but it reproduces.
Does it move to create maybe another?
I'm answering you. It doesn't move to form a duplicate or form a replicate. So it's even called a replicate here. But what it does, it reproduces to form a duplicate, and that is called binary fission. It's not movement. Moving is still the amoeba, but it will do something we call reproduction. Then it will split into two. Then we get to amoeba. But that one is the reproduction, so I will not explain that. But to answer your question, movement doesn't create duplicate. Then amoeba can form duplicates any day because anytime it is moving, then within a split second it can form about 10 duplicates. But the point is, moving is just to go away from bad stimuli and also get food to eat. So it moves to get food to eat. It moves to also live unfavorable environment. That is how it moves. But if you want to divide into two, form a duplicate, we call that one reproduction, specifically binary fission. So just be on the line. I will explain that better when we read that life. Now, Stephen, your hand is up.
Sir,
Yeah.
in the feeding process, does it undergo osmosis or what?
Feeding. Oh no. Feeding is just by phagocytosis. So it is getting the food. It's phagocytosis. So it depends on what is coming. If it's a solid food, it is by phagocytosis. If it is a dissolved thing, then it's by simple diffusion. But simple diffusion is actually more of the gaseous exchange. But the feeding is because normally the food could be particle. But if the amoeba is, let me say, lucky enough that in the environment, maybe glucose, maybe an organism fed, maybe a fungi was there, it fed cellularly, and there's some sugar over there, then those sugar can enter it by diffusion. But if it's a particle of food, that cannot be simple diffusion. It will be by phagocytosis, whereby it will, remember what we talk about that one in transport systems. That's the endocytosis. It will feed solid food by phagocytosis. And if there are nutrients like maybe dissolved sugar in the water, then lucky enough it can enter by simple diffusion. If you have a follow-up question, you can ask. Any other question on the feeding? Then I think I saw a question at the comment section. I'll go there. But if those who are ready to raise up their hand, any other hand that is up, you can come to explain.
Sir, please. That every curved part of the amoeba is called the pseudopod, or is that specific part?
Okay, I've understood your question. Let me explain to you. Now, any extension was once a pseudopodia or a pseudopodium. But normally, if you draw your amoeba, make sure one part is very, very extended more than the rest. So if I'm drawing my amoeba, I can decide to draw my amoeba this way. That is all. This will be my, how do you call it, the pseudopodia. So I don't have any trouble. Where's my pseudopodia? Then I'll be searching. But this one is to tell you that this amoeba was very, very active. Mine is less active. This one maybe a lot of food is there. So as I explained to you, It moved here once upon a time. That is the food that it got there. It moved this direction. It got the food there. It came here. That is the food it got. It went here sometime. That's the food. And the last point is here. The reason I'm saying that this one looks very close to the end, and that is where they even name. So any extension of the amoeba was once a pseudopodia or pseudopodium, but once it is done feeding and it is going to the next side, it will live there, but it doesn't live just fast. It's very very slow. So I've forgotten this animal. One animal is very very slow, the slowest animal in the world, and it feed on hyacinth. Like I've forgotten that name. It looks like a bear, very very slow. If the animal want to move, it moves slowly. It will be eating some bitter leaves. They say it's the laziest animal on earth. Amoeba is very slow than that. So this one was once a pseudopodia, which is now retracting, but it will take a long time. If it doesn't see any food here, it will become flat like this. Sorry, it will become something like this. So everything will be flattened up. So if I've drawn the amoeba, I would have drawn it this way, maybe something like this. So this one will not be there. This will be the only pseudopodia on the amoeba, and that is okay for me. So Lillian, any extension you see was once an amoeba, but if you want to label it, make sure to label the longest one to be the pseudopodia. If you name this one pseudopodia, I will not make you wrong because it was once. But if you name this part pseudopodia, you are wrong because this part there's no extension. They are all level up. But this is an extension. It is a curved, or let me say a projection that you have labeled. Any other question? Very good. Thank you very much. Someone said the animal, yes. The animal is a sloth. Thank you for helping me. So the animal is a sloth. SLOTH. And I've forgotten it feeds on some poisonous thing. No animal feeds on it. And that's the only thing that animal feed. Last time I was watching TikTok and said that it feeds on poisonous leaves. And even if you plug it and give it to it, it doesn't eat it. It wants to pluck it from the tree and take it. So that is the sloth for you. Very 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 slow. So you can search it on the internet. You will see how slow that animal is. So any other question? There was a question that someone asked about crystals. Yeah.
My question was that what is the function of the proteus in the cytoplasm?
Yeah, very good. I think that was where I was coming to. So our brother is asking the function of the crystals. The crystals, later on, we will talk about that. So someone can also do us a favor by searching. You can put it there. If there's a better explanation I can provide to, I will give to you. So we don't know what this crystal. It could be sugar crystals. It could be any crystal. So it depends on the crystal we are talking about. That can help us know the actual function. So Stephen, later on, if no one provides it here, I will do a quick search. When I'm uploading this video, I will include it over there for you. Thank you very much. The crystals depends. This one is urate crystals. So someone said it's a waste. So human beings, we see that our waste come as in a form of urine, and inside this urine we have what we saw the urea over there. Some organic, sometimes I don't know whether you sweat that way. You can sweat to a point that you will see, especially those who, if you swim, sometimes it could be gravels although, but there was a time I was working and I could taste that my sweat was very very tasty. If you are very dehydrated and you are working like that way, you can feel some crystals on your skin. It might not be sand. Those could be that instead of the urea coming out as liquid part, it can come as crystals. That is what human, if you don't take in much water, you can develop kidney stones. So but if you have more water in the system, it will come out as solid. So they are me, but the crystals, it could be urate crystal someone has put over there. So that's the waste storage. Remove essential. So that is the waste. So instead of them urinating, they don't urinate. Their waste will come out like the crystals. An example is the urate crystals from urea. So that's what Lillian has said for. I'm sure there will be a lot of these crystals over there. So the crystals depend on the type of crystal that we can talk about the fun. So the urate crystal, as is given by Lillian, help us to detoxify or take waste. That is, we try to urinate or excrete. That is it, and that maintain balance. Another one, someone said Yes, some also are for storage. So it could be oxalate. So the oxalate is for storage, but the urate from urea is for, let me say, excretion. So thank you very much. You've done a massive work. So it depends on the crystals. Some are just waste for excretion. Some are just for storage. Some could also be glucose crystals. Oxalate is also needed in the Krebs cycle. It's a sugar compound, 6 carbon sugar. That is a storage organ to regulate internal. So thank you very much. It depends on the type of the crystals. Some are good, some are bad that need to be excreted. Thank you very much. So let's continue. If there are no questions, I'll move to the next life process. So stay with quiet and Linian, thank you very much for bringing those information up for us. So the slow streaming movement that causes amoeba to move is called cyclosis. So keep this one. This has been a past question OBJ for several years. So the slow streaming motion that causes amoeba to move is called cyclosis. But it forms a projection that we call a pseudopodium or pseudopodia that allow it to move. So now let me do a quick read. So movement is by a slow streaming movement. Whenever I want to move, it pushes the endoplasm forward to form a pseudopodium or force field. So how does the endoplasm move forward? It can't just move forward in a vacuum. As it tries to move, what causes that the cytoplasmic content will move towards that direction. So it extends, it needs to be filled. So the cytoplasmic content will fill. So when you tickle that water, what happens is that that sachet water, the water will freely move to that end just to make it stand very strong or firmly. So that is what happens. So in the particular direction it wants to move, as it forms the new pseudopodium, previous pseudopodia are being withdrawn. Why are previous pseudopodia drawn? Because this is a new direction. So like if you put water on the table, so now if you have water around you, put water on your table. Now put your hand in the water and draw just maybe a line. You see that the water will be moving towards the direction that you do the line. So water will move from one end to the other side. If you do another one, the water will move from where it moved to go to the other side. So that is, if it is moving here, that pseudopodia there will withdraw because the the cytoplasmic content or the fluid is now being moved to that side, new direction. So it will move and it will withdraw. So previous pseudopodia are withdrawn. It moves by using the newly formed pseudopodia to move. The pseudopodia projects in the direction that the amoeba would like to move. Now, the movement and feeding is similar or the same. Now, why does the amoeba want to move? It is moving away from harm or it is moving towards food. So how does it feed? It feeds by phagocytosis. So the endoplasm extends to the direction of food. The cytoplasmic content moves towards that direction. This causes the extension of the cell membrane, forming a force we call pseudopodia. Now the pseudopodia moves around the food. It engulfs it and forms a food vacuum. Why are we calling the food vacuum? Food vacuum because now this is the food. It is in the water. The amoeba now extends the pseudopodia to cover it. Now it doesn't just cover the food. It covers the food together with water. Then that forms a food vacuum. So inside this vacuum, there's a cell membrane around it. There's the food inside and there is water around, which now the amoeba will now secrete digestive enzymes into it from the cytoplasm or from the crystals in it, and that will break down this food. Once it is being digested by simple diffusion, the nutrients digested, the glucose, will leave the food vacuole and now it will rupture or diffuse into the entire cytoplasm. Then the cell will begin to enjoy the finished food or the product from digestion. So let me come down to explain further. So amoeba is heterotrophic organism. Hetero means different. So we have two types of organism based on the feeding. Autotrophic. Auto means self-feeding. You can make your own food. These are the photoautotrophic and the organism that use chemicals. We call them chemoautotrophic. When I'm teaching nutrition, I'll go deeper. So for now, I'm just mentioning some terms for you, so that we don't spend much time. Sometimes it's good that I mention the terms for you to keep it. if I don't keep it, you might not be curious to study. So if the question become too much, if you have time, fine, I will explain. Don't worry. If you don't have enough time, I will just mention them. Keep them in your mind. Later on, you can be asked a question on the page or somewhere. So hetero means different. Trophic means feeding. We will learn what we call it ecosystem very very short in ecology, where we talk about trophic level or trophic feeding level. So hetero means different, trophic means feeding. So amoeba cannot make its own food. It depends on food from different sources. Therefore, it is heterotrophic. And it feeds on other smaller organisms. So there are some organisms that are smaller than amoeba. That's the bacteria, diatoms, and decayed remains of large plants. Amoeba is generally carnivorous. That means it feeds mostly on other animals, the bacteria and the rest. It doesn't feed on plant-like things, but it feeds on anything, but its appetite is largely for other animals, the bacteria and diatoms. And it feeds by phagocytosis. So Stephen, this answers your question. It feeds by phagocytosis by engulfing. It will the cell will extend and cover the food. It take you inside. That is it. So it feeds by phagocytosis. So it feeds by engulfing the food with its pseudopodia together with water, and that's from the food vacuum. I explained that. So the enzymes are then passed from the cytoplasm into the food vacuum to digest the food. After the digestion, the food nutrients will diffuse into the cytoplasm. So it will engulf the solid food by phagocytosis. Now the food vacuum become like its alimentary canal. After digestion, it will now be diffused as humans in our stomach or intestines. It will diffuse into the blood by simple diffusion. They too, after they've gotten the liquid glucose and the rest, then it will come by simple diffusion. But if the amoeba is too lucky to have dissolved glucose inside the water, then by simple diffusion, it just entered as water will enter. Then it enjoys its food. That is how amoeba feed. So the food serves as source of energy, you know already. It feeds because of energy so that it can continue its life processes. Now the undigested remains are ejected out of the body through the cell membrane. So that is called ejection, but it will just come out and that will become exocytosis. So keep that in mind. It will enter by endo, but particularly phagocytosis because it's a solid. If it was liquid, it will be pinocytosis. So keep those terms very well. That's why we learn those terms before the life process. Now, any question about nutrition, let's try and finish within today. For today, we will finish life process of amoeba. So let's try and finish today. Any question over there? If not, I will tackle the next life process. So we've talked about the movement and the feeding. Let's talk about respiration. So please, sorry if today we will go a little bit beyond our 9. I want us to finish. This shouldn't be 2 lessons, else the videos will be too much on the channel. So we are done with the nutrition. Let's talk about respiration. Now respiration, but also respire. That is by taking in oxygen and giving out carbon dioxide, and this is by simple diffusion. So that is all about is diffusion. So when this oxygen gets in, now it has glucose, it can enter its Krebs cycle on its own to get ATP out of the dissolved food nutrient. So amoeba undergo cellular respiration to produce energy. So it uses the oxygen. It has no special respiratory organs like we have the nose, the nostrils, we have the alveoli and the rest. For it, it is just the cell membrane to get in carbon dioxide gets out. That is all. So it dissolves through its general body surface, and we call that one plasma lemma. So the plasma membrane, the general body surface is called plasma lemma. That is like the cell membrane, the same thing, which is permeable to oxygen gases. So oxygen will enter, carbon dioxide will come out. That is how simple diffusion. So there's nothing serious over there. Now reproduction. That is what answers I said Chira Divine, or whether it was Patricia. Yeah, no, Patricia, just see your question. Amoeba doesn't move to duplicate or replicate, it reproduces. So amoeba also undergoes reproduction, and we call that reproduction acellular reproduction because only the amoeba will take part. Only one amoeba will undergo that. It doesn't need another amoeba. So one amoeba, when it is grown, it will withdraw all of its pseudopodia. So it will become like a stables, dormant. It will not be moving. It will withdraw all the pseudopodia and it will be something more like a circle. Now, it will first divide its nucleus. When the nucleus divide, they move to one end, another end, maybe north and south pole or east and west pole. Now, the cytoplasm will also divide, and together with that, they will split. So we call something karyokinesis. Then the cell will also divide. Then we have two different amoeba. Let me come. So we call that one binary fission. So remember, it's the two process we call binary fission. Binary means two. Fission means you are. So a fisher is a cat. We are dividing. Where the cell divide into two identical daughter cells. So it's like having one amoeba, you divide into two perfectly. There's no sexual reproduction in amoeba. So some books also say there are a form, but largely there is no form of sexual reproduction in amoeba. But some also try, some books and new information are trying to produce, but that has not been certified. So for now, and for our level of studies, we say that amoeba reproduce by asexual reproduction. When the amoeba reaches a definite size, as I explained, it withdraws all the pseudopodia, which the nucleus will lengthen and it will divide. Why are we dividing nucleus? Because nucleus is a critical part. It's like the CPU, the motherboard of the cell. It controls everything. So once you have a nucleus, you have life. You can just do anything on your own. So the nucleus will first divide, then they will into equal parts, they will move to at different side. Now the cytoplasm will divide into smaller manner. Now this happens only approximately every 2 days. So almost every 2 days amoeba will divide into two, and this depend on the species and the environmental conditions. If there are no enough food, amoeba cannot do that in 2 days. That's why feeding is important even for our health and our procreation. So during unfavorable conditions, if there is no food or the water is drying up, amoeba will withdraw all of its pseudopodia and become like a ball. It will form something we call a cyst. Now, I think it rained in Ghana, most parts of Ghana yesterday and somewhere even today. So before that, if you are very very vigilant, you could be seeing snails hidden around some point. If you pick a snail, I'm sure by now most of the snail will now be coming out by tomorrow, or some might even come out today. If you take up the snail, you see a whitish substance that covers the opening of it, the shell. We call that one also a cyst. Now if you put water on that thing, it softens, then the snail will come out. It does that to prevent death, because it is not raining and it cannot contain that dehydration. So the snail will now do that thing, form a cyst, and it will undergo what we call a hibernation, or it become a state of dormancy. The amoeba also does something like that. It will form a cyst, like a fluffy thread around its body. Then it will be there. It will move. It won't eat. It won't do anything. So it's like it is asleep. It's like putting your laptop or phone on sleep mode or hibernation. So the phone can do some simple things, but it will not do a lot of. So although it's on a sleep mode, the laptop time will be read, everything will be going, but it doesn't take a lot of your battery. So that is how the amoeba will form a cyst. And when it forms a cyst, it is prevented from drying up so that it doesn't die when conditions are unfavorable. So let me show you something here for the reproduction. So we see the amoeba has withdrawn all of its pseudopodia. Now look here, it looks very very circular. Now the cell membrane will divide into two. Now they will move to one side of it. Now the cytoplasm now becomes to divide into two. Then we have two different amoebae. Now any of them can move to wherever it wants. If they also get favorable conditions, they can also divide after 2 days, depending on the environmental condition and the species. But if these are not favorable, they will just stay dormant till condition become favorable. A hand just came up. That person can ask the question, or you can say whatever you want to say. The person that raised the hand, I didn't check the name, so kindly unmute your mic and talk. Okay, if that was a mistake, fine. So I'm continuing. A hand is up. Let me check the person. Cosmos Uswani, your hand is up. Yeah, Cosmos. I think this is the first time we are meeting here.
The cell division factor related to mitosis.
Okay, I get you. I get you. Okay, it's not related to mitosis, but it looks similar to that. It goes to the same process, but under mitosis, we are looking at DNA going a lot of things over there. So it's like, let me say, it's not mitosis, but it's a simple form of mitosis. Are you getting it? We call this one binary fission. This one is the entire cell dividing. But under mitosis, you see the chromosome will move. Are you getting it? There will be chromosome segregation, and so mitosis is an advanced form of what we are seeing here. Are you getting it? So if you want to liken it, this one will look like a lower version of mitosis rather than meiosis, but they are different things. Because here is the entire organism that is dividing. But under mitosis, we realize that DNA will be moving, just that there will be no crossing over. That's why it is mitosis, not meiosis. So they are different. Just that they might look a little bit similar, but they are very, very different. I don't know whether Cosmos you get my explanation very well. Yeah, so it's a very nice analogy that you did, but they are very, very different. Thank you very much. So we can continue. At least he has gotten the concept and he can now relate it to mitosis. Okay, let me talk about excretion. If there are no questions now, I'm getting some feed. Is someone microphones on? Kindly. Okay, sorry, Cosmo. Let me mute you and see. Okay, at because I have a lot of devices around me. If you also open your microphone, I get a feed around the devices, and that will not give me a good recording for editing for the YouTube page. All right, so let me talk about. Sorry, we are almost around 1 hour 30 minutes. So today I've apologized. Let's try and finish everything. So let's hang in there and finish everything today, so that next week Sunday we can talk about the next organized euglena. Yeah. So now, excretion. We have the contractile vacuole that serves as water storage. That is what it's used for, excretion. So waste products are continually removed from the cytoplasm of the amoeba. Now, the more contractile vacuole are formed, that means the amoeba is in a hypotonic solution. So more water get into it. So if more water is getting into it will have more water vacuole. So if it is in an isotonic solution, it will have lesser contractile vacuole. And if it's in a hypertonic solution, then the amoeba might not even form a water vacuole because it will not water will not be moving frequently in it. So waste products are continually removed from the cytoplasm through the cell membrane. And this is what happened. The waste product dissolved in the amoeba will enter the amoeba through the cell membrane. The water in all these things will now form a small bubble that we call a contractile vacuole. So the contractile is like a bubble with the water itself and other dissolved organisms, so other dissolved substances. So more water with water products accumulates in the contractile vacuole. So any water or waste product will now be found in the contractile vacuole. This will cause it to increase in size. So the contractile vacuole is like very good. Sometimes you can see bubbles in water, or even when you put a fizzy drink in a cup, you see that sometimes these bubbles can meet. Very good example is when you are having a soapy water. Sometimes you see you can join two bubbles, or as you try to shake it together, you see that one small bubble can meet a bigger one. Then it meet together, it form a very big bubble. Sometimes you can see another one overlapping. So you can be doing that one. As case we did a lot of these things. A soapy water, you just be playing with it. You see that the bubbles can meet together and they form a larger one. So that is how these things happen. Then once they clamp together, or even your phone screen, if you are not good at placing the the protector over there, you see some air bubbles in between the protector and your phone screen. Now you can move them together and they form a larger bubble. Then you can just push it out of the screen, then all the bubbles go off. So that is what happens in a contractile vacuum. They pick up small thing, then it become larger. When it become too large, it also move closer to the cell membrane. So let me show you something here. When the cell membrane, when the contractile vacuole is full, it will also move to the end of the cell. That's the cell. So this is the contractile vacuole. So now check where I'm using red. Let me use red as my pointer over here. So now this is the contractile vacuole. I'm trying to draw the outline. Now when it is full, you see that this is the cell membrane. So let's assume this pseudopodia has now withdrawn. So the pseudopodia will now withdraw. It will become something flat-like. So this will become, this will become the amoeba's cell. So that's the amoeba. Now this contractile vacuole has now pushed to the end of the cell membrane. Now let me change the color of this so that you can see it better. What happens is that the cell membrane here will now withdraw. So this cell membrane will now withdraw. Here too will withdraw. Then it will cover car outside. This one will now move out. So what we have here as the contractile vacuole will now move out of the cell. So now it has come out of the body of the cell. Once it is out, the cell membrane will now lengthen again like this. So now everything is off. Let me do it here again for those who don't understand. Let's assume this is my contractile vacuole. So this is my contractile vacuole. Once it is becoming bigger, so it will pick up smaller contractile vacuole. They will pick up together. It will become something big like this. So when it becomes bigger like this, it will move to the end of the cell membrane. So it will come somewhere here. Now what happens is that the cell membrane will now withdraw inward. So when the cell membrane withdraws inward, it will push this one outside. So the cell membrane now becomes something like this. It will now form something like this outside of it. Now this one now will move out of the cell membrane. Now my amoeba will become something like this with this one outside. So that is how it undergoes what we call the excretion. So the contractile vacuole, when it is full, it moves to the end of the cell membrane. Now the cell membrane moves inward, now causing the contractile vacuole to move outside the cell membrane in a process called exocytosis. So when that happens, we say the contractile vacuole has now splitted, or it has burst out of the cell membrane and it has been excreted or taken off from the cell. So excess water will go. If there are some waste, it go. If the water too is very less in it, then it will be only the crystals that are the urate crystals and some other waste substances that will be forced to move out. So let me read something here for better understanding if someone still find it difficult to look at the demonstration. So finally, so the more water products accumulates in the contractile vacuole, it will cause it to increase in size. When it becomes too bigger for it to burst, it moves close to the cell membrane and it will burst. So once it bursts, it expels its content outside the amoeba. Waste material like carbon dioxide, ammonia escape by simple diffusion to the cytoplasm, to the ectoplasm, and water balance is maintained by osmoregulation. So if you are very thirsty, your urine become too small or very scanty. If you take in much water, you have a larger volume of urine because more water is in your body. So that happens to the amoeba. It controls water regulation. Your kidney also has osmoregulation. If you have a lot of water in your body, you have a very clear urine, not yellowish, and very voluminous. If you have less water, you're not drinking water, your urine become a little bit slothy and also yellowish and very small. So and salty as well. Now osmoregulation. I explained this at the start. Osmo means water. So regulation means water regulation. So it's the process whereby organisms like the proteus and organisms regulate water in their body. Humans also does that. So all organisms does, but we don't use control that. We use our kidney, our sweating, and our skin for doing a lot of these things, and even the blood movement of water in the blood. Sorry. So osmoregulation is the process of regulating amount of water in the body of organism The cell membrane of the amoeba is semi-permeable, so it allows water to be taken or removed as a result of osmotic pressure in and out of the cytoplasm. So it is done by the contractile vacuole. When it reaches a certain size, it will come closer to the cell, it will burst and release everything out, or it will be exocytosized. So this will contract, forcing water out of the cell. So this is example of osmoregulation. So osmoregulation is like the reverse of feeding. So feeding, it will move inside, but excretion, it is coming out. So it's just like the reverse. So water enters the amoeba, it will become larger. Contractile vacuole swells up. You see, contractile vacuole has come to the end here. Now, when it comes closer to the cell membrane, it will now burst and everything will come out. It will start the process again. Now, there's a small water bubble. It will now become larger like this. It become larger. When it's too large, it comes outside. Then it comes out. It will form another one again. Then you control the cycle on and on. If it is still in a very hypotonic solution, a lot of contractile vascule can be formed. Now, response to stimuli. How does amoeba respond to changes in the environment? It responds by moving to favorable ones and moving away from the unfavorable ones. So once is a very good something like food, it moves towards that. So and it moves very very slow, still with the pseudopodia and cyclosis movement. So this is so amoeba such as moving towards the food source or away from harmful substances. They have the simplest cell signaling pattern, so they don't have a complex how do we call it neural arc or neuron that will help in this pathway for sensory perception. Just a simple system for signaling. So that is chemicals, light, heat, and food that it moves. Now, anytime we learn an organism, we talk about adaptation. So sorry, we are still late into it, but let's finish it so that it becomes. When we talk about adaptation, we have structural adaptation and the rest. When we just say adaptation, it means anything that the organism have or any behavior that it can do to help it live. When we say structural adaptation, this is very important in practical. Structure refers to what it has, like the body part, and we have behavioral adaptation. What does it do? But when we say adaptation, straight like that, it could be structural or behavioral. So this one is very very free. Adaptation, you can write anything. So it has contractile vacuole that helps it in regulating water or for excretion or in osmoregulation. It has a flexible cell membrane that enables it to change its shape or can move by pseudopodia. You can talk about it has a large surface area that facilitates simple diffusion. So carbon dioxide can come in. How do you call it? Carbon dioxide can move out, oxygen can move in easily, and also diffusion of the dissolved nutrients into the cytoplasm. Now, it has a thin outer membrane which facilitates simple diffusion, and you can talk about the cell membrane which is very flexible that enable it to form pseudopodia. These are the structures in amoeba that enable it to perform its life process or enable it to live. So when we say adaptation, is any feature or structure of the organism or behavior that allow it to thrive or perform its life processes. So that is the adaptation of amoeba proteus. Now, encystment in amoeba. This is just trying to explain the encystment. That is where the conditions are not favorable. Most especially when you are hungry, you are very quiet. Why are you quiet? You are conserving energy. The day that you be fasting, you see that you are very quiet. You don't play football, you don't be talking, you don't talk a lot in class. You are very quiet. Even as you person cry, you don't even want to talk because you are conserving energy, you are hungry. But after eating, that is when you are very energetic. You can move from one end of the class to the other. So when the amoeba is under unfavorable condition, there's no food or there's a drought, they also try to form what we call a cyst. And we call that term also as sporulation. So let me explain that one. So they form a cyst, and the cyst is the protective covering that covers the amoeba. So the protoplasm within the cyst divides repeatedly into many young amoeba. So the amoeba is under that stage. It will also undergo reproduction at that side, and we call that one sporulation. So aside binary fission is when things are okay with it. It will just divide into two. But when it is undergoing what we call the sporulation, it is at the point whereby the organism is undergoing the encystment. So inside it, it will still be uh, dividing, so it can divide repetitive into many young amoebae. When this happens and condition become favorable, the cyst can now break. When water touches it, it now dissolve, it now open up, and we can get a new young amoebae that will come out as the new organism. So we call that one sporulation. But on the face, when things are okay, it undergoes binary fission. When things are not okay, it can undergo what we call sporulation. So that is the encystment in amoeba. Now, economic importance of amoeba, and we are done. So is economic importance in biology. Economic importance could be good or bad. So whatever it does, if it is good or bad to you, that is its importance in the ecosystem. So they assist in nutrient recycling because of how they feed. So what they do is that now they feed, they also serve as food for other organisms. So they also help in that, such as bacteria and algae, to maintain ecological balance. So they feed on bacteria. If the bacteria is a bad one, they are doing good to us. If it's a good one too, they are causing harm, but still is an importance in the ecosystem. And they serve as model organisms. So now we are studying about amoeba. They serve as model organisms in scientific research. So that's what the importance. So we have people that study them, and they play a key role. They form an important component of the food chain. So in the food chain, they are also heterotrophic feeders. So they feed on other organisms. They also serve as food for some organisms. So humans will not feed on amoeba, but indirectly we feed an organism that feed on amoeba, which if they don't get food, they cannot supply for us. So that is the chain, or the food chain, or the food web. They play important role in maintaining a stable ecosystem. Now they serve as indicator species in habitat. The reason is, as they change the number of populations, are an indication of interference of water quality. We remember that they are found in clean water. So the more polluted the water becomes, the amoeba now die out of the pollution, or out of those activities. So the more amoeba we get, it means the more clearer, or let me say pure, that water will be. If the waste accumulate, most of them will perish or die as a result. But some amoeba are also parasitic, some are pathogenic, but most of them are free living. So we call something entamoeba histolytica. So this is an amoeba that is in a way harmful, and it's called what we call dysentery. So it's called amoebic dysentery. So entamoeba histolytica is an example of amoeba that is harmful, and that can cause dysentery in humans. So some fish are parasitic, some can damage crops. For example, is the entamoeba histolytica, which will cause amoebic dysentery. So I remember someone asked a question some years ago, and they do ask in class. I'm sure someone would have asked, is amoeba harmful? After knowing bacteria, some are harmful and some are good. They want to know whether amoeba to some are harmful. So amoeba, naturally, the amoeba that we are studying is harmless to us, but a species or strain of it is harmful. Example is the entamoeba histolytica that can cause amoebic dysentery in humans. Now we are done with the life processes structure of amoeba proteus. If there are some questions you can ask, sorry today we have gone beyond our time because we wanted to finish amoeba today. If there are some questions that we haven't tackled, you can ask. We are waiting for just 2 minutes. By 9:16 we are ending the lesson. If there are no questions, you can ask your questions if you do have one. Okay, thanks for understanding. I've seen some thumbs up. It means they do understand, they appreciate. Any other, you can raise up your hand. I'm waiting for the next 20 seconds if there are no questions. Okay, do all cells in animals respire by oxygen? Okay, yes, most organisms, let me say all, we need oxygen for respiration. So for respiration, remember, respiration, we need oxygen. but there are some other organisms that can do without it momentarily. So we have instead of carbon dioxide and heat, sorry, some organisms are because organisms methanogens, that's the green sulfur bacteria, they can use methane instead of chemicals for food production. The fact that they are using the methane and carbon dioxide for food production does not mean that after the food production they will not need oxygen for ATP. So all organisms, if only they respire, will need oxygen for acellular respiration. But remember, we have how do you call it? Sorry, for aerobic respiration, not acellular, aerobic respiration. We have some organ that can undergo anaerobic respiration. So some can do without oxygen. That is when it is not there. So we have organisms that are anaerobes. So we have anaerobic bacteria. They can do without oxygen. It doesn't mean that when they get oxygen, they will not do without it. So it's like now we don't have a car, so you take trotro, you take motor, or any, or you work by foot. It doesn't mean that when there is no car you can't. But some people that are used to the car, the day their car breaks down, they cannot do away with it. So they have become what we call the aerobic, they need it. But the anaerobes, they can do without the oxygen. But when there's an oxygen there, they will do better with it. So thanks for the questions and requirement. So we need oxygen, but some can do away with oxygen under anaerobic respiration. Any other question? So I think that is all. Okay, thank you very much for joining tonight's lesson and making it a success. So Stephen Quarton, Divine, Patricia, Linian, and everyone that made a success today. I saw some new names. I think I've forgotten the names though, but you've all done very very well for joining for tonight. Yeah, Zadabi, that was the name I was looking for. I think Zadabi, Bright Sarah, and a lot of you joined. So I appreciate you. Have a very good night. God willing, on Sunday we will talk about the life process of euglena. So please keep on studying, not just biology, learn the other subjects as well. And until then, keep studying. So I'll make sure this video becomes available latest by Thursday on my YouTube channel. You watch, share with your friends, subscribe if you haven't done as at now, and I'm sure we will do a lot of studies to impart knowledge onto you. Thank you very much once again. Have a very good night. Bye to you all.